our HERN. Thank you all for joining our HERN webinar series. Today, I will be introducing a couple of excellent speakers. I'm Joyce Nyland from the City of Hope. I'm Principal Investigator of the Coordinating Center for the Human Islet Research Network, or HERN. And today we have Dr. Rohit Kulkarni and Dr. Dario De Jesus from the Joslin Diabetes Center. I'm really looking forward to their presentation on mRNA modifications in islet biology. Let me just give you a brief overview of what HERN is and a few opportunities you have currently to possibly join HERN. We began HERN in 2014. It now consists of 5 consortia plus the Coordinating Center. There's the CBDS Consortium on Beta Cell Death and Survival, CMI Consortium on Modeling Autoimmune Interactions, CHIB is the Consortium on Human Islet Biomimetics, CTAR, the Consortium on Targeting and Regeneration, and then HPAC is the most recent, the Human Pancreas Analysis Consortium. These 5 consortia form the network. It's a very fluid network that has evolved over time and will continue to evolve. It's NIH, NIDDK funded. And the mission of HERN is to better understand how human beta cells are lost in type 1 diabetes and to find innovative strategies to protect or replace the functional beta cell mass in diabetic patients. We serve as the Coordinating Center at City of Hope. It's called the Human Islet Research Enhancement Center, or HIREC. So there are now over 200 HERN investigators that have been funded over the years, involving 85 institutions and spanning 9 countries. So it's definitely international. If you go to our HERN website, you can find a lot of information there, some of which I'll show you right now. Job opportunities, funding opportunities are there too. And there are 3 out right now that I just wanted to briefly highlight in case any of you are interested in applying. So one is the High Resolution Exploration of Human Islet Tissue Environment. This is a UO1 from NIDDK to contribute to a higher resolution understanding of organization of human pancreatic tissue environment. Multidisciplinary teams are encouraged, and whoever successfully applies to this RFA will become part of the HPAC, the Human Pancreas Analysis Consortium of HERN. Deadline is March 3rd of this year, budget up to $600,000, direct costs for 4 years. So a very nice opportunity to get involved with HERN. Then there's the NIH Gateway Award. It's not directly involved with HERN, but it includes HERN as one option. This is designed to support robust pipeline of innovative projects and talented new investigators in T1D research. It's aimed towards early stage investigators, providing support prior to their first R01 submissions. This is an R03 mechanism. Whoever succeeds will join one of 3 collaborative NIDDK networks, either HERN or TrialNet or TEDdy. And the deadlines here, there's 2 funding rounds, June 1 of this year and March 20 of next year for $100,000 direct costs for 2 years. So you might tell your new investigators about this opportunity. And then one more funding opportunity is directly funded through the opportunity pool of HERN, and we manage this through our HIREC. It's called the HERN Catalyst Award. Here we're looking for proposals that are bold, innovative, challenging, and reflect new insider understanding to the field. Has to be an original idea, not continuation of currently funded work. And it would need to fall within the purview of one of those consortia that I showed you a moment ago. This one is due May 2nd of this year. The budget will be $125,000 direct costs up to 2 years. So again, we encourage you to check these out and apply to any that are applicable to you. When you join HERN, a key requirement that the RFA has from the NIH is sharing of resources. And we're going to highlight that today. The methods, reagents, resources, biomaterials that you develop, her protocols, models, they all are expected to be made public to the research community. And you need to really adhere to this and share your resources. So that's why we set up this webinar series as well, so we could share with everyone in the diabetes community. We're happy to do that. And the HPAP program, which is under the Human Pancreas Analysis Consortium, is set up as specifically a resource generation program that gets pancreatic samples and analyzes, distributes high-value T1 and T2 data sets to the diabetes community. So if you want to go there, this is called PancDB. It's part of the HPAC of HERN. We in the HIREC have set up the HERN resource browser, and we're going to show you how to access this today. It has now over 1,400 entries cataloged in these various categories that you see here. So you can get protocols, you can find out about mouse strains, data, antibodies, cell lines are all there. So we encourage you, this is available to anyone in diabetes research. And we also want you to know that the videos of past webinars like today are posted online at our website as well. So you can go and look at any past videos, and today's is being recorded and will be available for you and your colleagues after today's session. So let me just briefly outline today's webinar. And today's presentation will include an introduction to the field of RNA modifications, decoding the functions of M6A mRNA methylation, sequencing strategies such as EMRA-seq, differential analysis, useful online tools and data sets, and then applications in the study of type 1 and type 2 diabetes. Then they will talk about emerging concepts and challenges in the field. We'll save our discussion to the end. Feel free to put any of your comments or questions in the chat, or you can wait till the end and you'll be unmuted and you'll be able to interact with our speakers for today. And with that, I'd like to pass it off to Rohit. Dr. Kulkarni, would you like to begin? I will stop sharing. And if you could share your slides, I'd appreciate it.
Thank you very much, Joyce. Just want to confirm you guys can see this?
Yes, that looks good.
Great. Thank you, Joyce, Leila, and John, and the HIREC for organizing this very timely webinar. And we're very excited to share some information that we have learned over the years. So let me begin by giving you an introduction to this topic. And just want to emphasize that we won't be covering too much of the detail in this, but we'll leave that to the discussion, but just give you enough overview to make you excited to get interested in this area. So I'm going to give you a little bit of the introduction to the field of RNA modifications, and then follow that up with a little bit more detail. So this is just a cartoon to show the different types of RNA which are important. As you can see, many of our colleagues at HERN and outside, like Laurie Sussel, Yuval Dor, Guy Rutter, to name a few, have been interested in many aspects of RNA biology. But my lab has been interested in mRNA, and that's the topic that I'm going to focus on for the next few slides. So just to remind you a little bit of biochemistry, the pyrimidines and purines, I'm sure you remember you have adenine and guanine, which are the purines, and you add the sugar moiety to this to form adenosine. And what we're talking about in the context of RNA modification is when you add the methyl group to the adenosine moiety. And so why is this important and how did this come about? First point I want to make is that it's extremely conserved, and there are a number of modifications of mRNA shown in the common Venn diagram here. I'll go into the acronyms a little bit in detail, but just to emphasize that this tells us that this is highly important in biology. So just an overview about the different types of modifications. So shown on the top is the cap region, the 5' UTR, the coding region, and the 3' UTR. And below that is representing the different modifications shown in the different colored circles. The size of the circle represents the amount of modification in that region. And you can see that M6A, for example, is got a large amount in the 3' UTR, as well as the coding region, and as well as the 5' UTR. But you can see that there are so many modifications which are present, which tells us that we have underappreciated the role of RNA biology quite a bit in metabolism, although much of this has been studied in cancer biology. So the one that we are interested in is M6A, and I'll go into a little bit more detail there. But I just want to also emphasize that M6A is not the only modification, and many of us are now realizing that other modifications like pseudourylation, there's another modification that we're recently become interested in, is also important in RNA biology, as well as 2' methylation. So just to keep in mind that many of these modifications are critical for various aspects of transcript biology. So just one historical point here, shown here on the left is Francis Crick's first lecture, The Central Dogma, where you have DNA, RNA, and protein. And there was some thought at that point that the DNA goes all the way towards protein, and the reverse is not possible. But there were some possibilities discussed at that point where RNA could regulate DNA. And so many of these modifications didn't come into light until 1974, when Fritz Rothman made the first discovery of methylated nucleosides in mRNA from hepatoma cells. This was immediately followed by another group in 1975. But it took us about 30 to 40 years to develop the methodology, and you'll find that the first initial observation began to emerge in 2012, 14, 16, and onwards, when the mass spec approaches became more sensitive to really interrogate and quantify this methylation. So this is not a really new topic, but just tells us that it's challenging, and we have to wait until methodology is available to interrogate these modifications in different aspects of biology. So a couple of other examples here where you have comprehensive analysis of mRNA methylation, again confirming its enrichment in the 3' UTR and the stop codons, and then finally comparing the human and mouse M6A RNA methylomes by sequencing approaches. So one other point I wanted to emphasize that got interested in this is many of us are aware of modifications of DNA on the one hand, where you have DNA methylation, and also histone methylation or acetylation, which indicates modifications of protein. And we always thought there was something missing. And then when Dario came to the lab several years ago, interrogating the role of insulin IGF-1 signaling in the beta cell, he came up with the comment that it's very surprising that metabolism, we hardly do any work on the RNA component, which may still be important in looking at epigenetics, and so the new term epitranscriptomics. So that the interest about reversible RNA methylation, which can have an impact on DNA methylation on the one hand, as well as histone methylation on the other. And therefore, we think that RNA is an important player in epigenetics. So what exactly are we talking about? Just a little bit of introduction here. So typically with the transcripts, when they get methylated, it all starts in the nucleus, and they're driven by an M6A writer complex, and it is a complex. You can see the large number of proteins which are involved here. Shown here on the cartoon on the left are just the two of the important ones, metal 3, which is considered a catalytic subunit, and metal 14, which is the other player, along with metal 3, which in addition to WTAP and a host of other proteins, forms the writer complex. So without going into great detail about the writer complex, just to emphasize that the methylation is added on to the transcript in a co-transcriptional manner by the writer complex. And as the transcript continues to traverse through the nucleus, it then gets exposed to what are called as erasers, chief among them being ALPH5. But you also have an eraser, the FTO, which you might have heard about, which made waves a few years ago in a paper in Nature, But the FTO is an eraser which is considered to be associated not chiefly with M6A, but the M6 dimethylation, so M6AM, and mostly associated with small nuclear RNAs. So as the transcript then progresses to exit through the cytoplasm, it then gets recognized by readers, for example, YSDC1, which is involved in splicing as well as export. So the transcript then could be decided that the transcript gets exported to the cytoplasm, where again it gets recognized by additional set of readers, the DF family of readers, as well as IGF-2BP1 and FMRP and others. And this is where the transcript then gets either degraded, translated, and therefore the fate of the transcript is in the hands of the readers, and these readers are recognizing the methylation pattern of the transcripts. So you can see it's a very well thought out process on the transcripts, and telling us it's pretty important. One advantage in this context is that it is conserved between mouse and man. And so here's an example of the zinc transporter showing how similar it is between mouse and man, and this is the consensus motif for the track sequence. One other point to make is that the methylation that we are interested in is in mammals is because it has a large number of methylation on adenines, up to 0.4% of adenines in RNA, and we see as many as 5 M6A sites per mRNA. So approximately 7,000 human genes out of the 20 to 25,000 have mRNA which are methylated, and up to 300 non-coding RNAs are also methylated. So that gives us some kind of perspective as to why we need to be interested in this. So at this point, I think I will hand it over to a bright young postdoc in the lab, Dario, who will tell us a little bit more about the details. Thank you.
Thank you, Rohit. Hello, everyone. We now know that M6A is extremely dynamic. We know that M6A mRNA modifications are abundant, are reversible. And the questions are, how do we map it and how do we analyze this data? In the last couple of years, we have witnessed a true gold rush towards the development of new sequencing methods, new protocols that basically focus on improving this in two main aspects: in decreasing the amount of required RNA, and by improving the resolution of the sequencing, ideally bring it back down to one single nucleotide base resolution. I will just show you two examples very briefly. Such an example is the M6A CLIP sequencing. And here, by UV cross-linking an anti-M6A monoclonal antibody to RNA, we are able to decrease the resolution to one nucleotide and to bring down the initial material requirements to 3 micrograms of poly-A sequencing. There are some limitations of using antibodies to perform these IPs, and so another recent method is the MasterSeq, is the antibody-free method, in which they take advantage of MASF, methylation-sensitive ribonuclease that will cleave every single methylated adenosine, and then by next-generation sequencing, we can map the M6A sites. And one good aspect of this technique is that we can bring down the amount of RNA that we need to perform this to 100 nanograms of poly-A RNA. However, the true gold standard method to map M6A is still the M6A sequencing, also known as MRIPSeq. And I will just describe very briefly some of the steps that we take to perform these experiments. So we start by isolating mRNA. we can isolate mRNA these days directly from cell lines or tissues, but one can also purify mRNA from total RNA. We then have to fragment our mRNA in small fragments with 50 nucleotide or 100 nucleotide of length. And then we're gonna use between 95 to 98% of our mRNA to perform an IP using a monoclonal antibody anti-LAMP6A. And then we're gonna use the remaining 2 to 5% of mRNA as our input sample. The next step relies on the utilization of specific low input RNA sequencing library kits. And then we need some specific peak calling software to analyze our data sets. And this is the typical data that we get. We have the read coverages. Here I'm showing an hypothetical gene where we have the input in black, and we have our IP sample here in red. And so these methods allow us to have an idea about the different proportions, the different areas of peaks, and they allow us to map and see where these maps are in the gene structure. Well, how do we do this analysis? How do we do peak calling? And why do we need specific methods, specific packages for peak calling when we are working in RNA modifications? Well, the main reason why we need specific packages for RNA is because, contrary to DNA methylation, we might have very strong changes in our input, in our basal RNA levels between a treated condition and a control condition. So we can find, and there are several packages that we routinely use for DNA methylation analysis, but these don't accumulate this RNA heterogeneity, and so we needed specific packages for this. One peak calling software that we really like is the QNB, and it uses a negative binomial model. On the top right panel, I'm just showing you the area under the curve of a ROC curve. So a ROC curve is just a normal, easy way that one can measure the specificity or sensitivity of a test or a biomarker and so on. And on the x-axis, we have the replicates, and on the different colors, you can appreciate the different peak calling methods being compared, with the QNB here in pink. Well, the first observation is obvious, right? How the sensitivity and specificity of the different tests increase when we increase our biological replicates. But the second thing one can conclude for this is when you look at the QNB almost with many different numbers of biological replicates presents increased specificity and sensitivity compared to the other peak calling software. So this is really a package that we use when we have smaller numbers, or when we have a maximum of two conditions, treated versus untreated, or knockout versus scrambled. And it's usually a peak calling software that we use when we don't have covariates. So we often use this when we are performing experiments in cell lines and we have an N equal to 3. Well, but with our human analytes, we had a big problem, right? And is a problem that is shared with everyone working with human complex diseases. And the problem was that our human analytes were full with covariates. So different genders, different BMIs, different AOCs, different viability, different purity, and so on. So Scott, which was a super talented PhD student in the lab of Professor Shuan He, and now he's assistant professor at the Yunnan University in China, he took care of these limitations, and he really built up a new peak calling software that uses gene level read count to normalize our input level instead of using peak level read count. And another difference of radar is instead of using binomial model, it uses Poisson random model. And so these allow us to accumulate for covariates such as age, BMI. On the right panel here, I'm showing you 4 different data sets, so you can appreciate how radar really performs better than other peak calling packages. We have on the lower 2 mouse data sets, mouse liver, mouse brain. On the top, we have 2 human data sets, ovarian cancer and our type 2 diabetic human islets. And on the y-axis, you have these histograms. So on the y-axis, you just have the number of genes, and on the x-axis, you can appreciate just the p-values. So when you look at the red bars, you can see that radar really provides us with the highest number of genes with really low p-values compared to all the other packages. A limitation of some of these peak calling softwares, it might be that they might generate false positives. So a way that we can control for this is by premuting our samples, is by relabeling and mixing our samples. And so if what we are seeing is really false positives, then whatever you've mixed your samples, you should still see the false positives. And here in purple, you can see that when you permute our samples, we totally lose these.
Have you done this before?
These number of
highly
significant genes. And this is not true particularly for packages like Fisher or ExomePeak packages. But radar had also limitations. And a main limitation was the previous software. You only allow us to control for 4 covariates. So how great would it be to have improved radar in which we can control for many more covariates? And how cool would it be to have a package in which it could run in parallel radar and QNB? Well, Scott, again, he took care of that problem, and he just wrote a new tool. And this is the tool that we are currently using in the lab, MRIP tools, and everyone can have access to it and download it. So I show you how we map M6A, how do we analyze M6A data. But on a monthly basis, we get emails asking us, can you please check if this candidate gene that I'm interested is methylated in your data sets, if this gene changes when you knock down M3 or M14 in beta cells. And we love to get these emails. We love to reply to these emails, and we love to have people reading our papers. But you can find useful online tools that can provide us with some idea regarding if M6A is binding or regulate your gene of interest. A package that we really like is the M6A2 target. The website has a very simple layout. You just have to input your gene of interest over here and you press go. And then you will have to select which cell line and which system you are interested in. And I'm just showing an example of MAFE, a gene that islet biologists all know. And I'm focusing on, and I'm selected here, HeLa cells, the human HeLa cell line. You can see here by looking at the binding that people have already done this experiment, and they have shown that the M6A reader proteins YTF1 and 3 directly bind to MAFE. You can also see, for example, that when we look at the perturbation, that when people knock down methyltransferase 14, an M6A writer, you have a change in the MAFE gene expression. So this is like a very useful tool that can give you an initial idea about if your gene is recognized by M6A writers or erasers, and if you should expect any change in gene expression of your gene of interest. Another very useful data set is another very useful resource is the Earn Resource Browser. And here, and I invite you all to take a look at it. And here we have uploaded a couple of data sets. So here you can find RNA-seq, so our input sample, our input data, and our M6A sequencing data in human type 2 diabetics compared to controls. You can also find RNA-seq and M6A-seq performed in endo-CBH1 cells, a human beta cell line, where we knock down methyltransferase 3 or methyltransferase 14, two M6A writers. And you can finally see an RNA-seq data set where we sorted beta cells from methyltransferase 14 knockout mice, and we compare it to control flux mice. So you can freely download and reanalyze these data sets, and there is a lot of data here, and that might be very useful to some of your projects. So how do we apply this? Like Dr. Kulkarni said, in the lab we have an interest for a long time in really understanding the beta cell dysfunction associated with type 2 diabetes. And we also realized that, especially by looking into single cell RNA-seq data performed in type 2 beta cells compared to non-diabetic patients, beta cells, we detect a very small number of statistically significant genes. I think some people quantify this, and less than 0.6% of all the detected genes change in type 2 beta cells compared to non-diabetic. So we really thought that there must be a new layer of regulation that is in between DNA, RNA, and protein, that is between RNA and type 2 diabetes disease. And we really thought that M6A would be such a layer. We started, I'm showing you very briefly some published data. We started this by downloading and reanalyzing a very known single cell RNA-seq performed in type 2 diabetic islets. I'm showing here the beta cell data where I'm comparing controls in red versus the type 2 beta cells in blue, and I'm showing different M6A modulators. You can appreciate how the M6A writer methyltransferase 3 and how M6A eraser LKBH5 and the M6A reader protein YTHDF1 are both downregulated in the type 2 diabetic beta cells compared to controls. Well, we validated these by ourselves by really getting access to human islets, culturing them overnight, and then just performing a couple of qPCRs. And again, we could see that by comparing the human type 2 diabetic islets seen here in red to controls plotted here in black, we could see that indeed methyltransferase 3, LKBH5, and several M6A reader proteins were downregulated in T2D beta cells. We further validated this at protein level. We got islets from type 2 diabetic patients and control patients. And again, methyltransferase 3 and M14 were downregulated in T2D compared to controls. And very recently, there have been a couple of publications showing exactly the same thing, showing that M6A writers are decreased in T2D and that high glucose levels indeed downregulate methyltransferase 3 and 14. So we decided to go ahead and perform M6A sequencing. We cultured 7 independent human islet preparations from control patients and 8 T2D islet preparations, and then we performed sequencing. I'm showing here our input data, so our normal RNA-seq data. You have a PCA on the left where the T2D are in blue and the controls are in red, and you have a heat map of the top 50 beans here on the right. You can see that indeed the control samples, they segregate from the T2D, but you can see how they sometimes overlap and how there is a lot of variability in the controls as well as in the T2D human islets. We were amazed to see that when we look at the M6A data, the type 2 diabetic human islets clearly segregated from the controls. And most importantly, the T2D human islets were very homogeneous compared to the control human islets. We did a gene ontology in the differentially methylated genes between T2D and controls, and we could see enrichment for cell cycle, insulin signaling, and pancreas development. We then digged a little bit more into these genes, and we found several hypomethylated genes belonging to the insulin and IGF-1, AKT, and PDX1 pathway. And I'm just showing here 3 examples of it. IGF-1 receptor, PP3CA, which is an AKT phosphatase, and an old friend of islet biologist, PDX1. So all these genes involved in this pathway presented hypomethylation. We published this in 2019, and so we showed here in this paper that diabetes indeed impacts the expression of M6A modulators in human beta cells, and that M6A decorates genes that are fundamental for beta cell identity and function, including MAFE, PDX1, and several other Moody genes, and that methyltransferase 14 is fundamental for beta cell mass maintenance and proliferation. When we were analyzing one of the last pieces of data from our previous project, the RNA-seq that we performed in the sorted beta cells from the methyltransferase 14 knockout mice, we saw several pathways associated with immunity, innate immunity, and antigen presentation. So the next project to us was very obvious. It was to study what is the role of M6A in type 1 diabetes. We started checking this by isolating mouse islets from NOR mice, a mouse model that does not show insulitis, and NOD mice with 4 or 8 weeks of age. We cultured them overnight, we isolated RNA, and then we performed qPCR. I'm showing here the NODs in pink, in red, and the NOR mice in gray, 4-week-old mice. And you can see that in the early stages of diabetes, at 4 weeks of age, methyltransferase 14 and methyltransferase 3 show an upregulation compared to NOR mice. But when we look at the mouse islets from the 8-week-old mice, we see that the NOD mice compared to the NOR mice presents a downregulation of methyltransferase 3 and M14. We further validated this, so we isolated pancreases from NODs with 1, 2, 4, or 6 months of age, and I'm showing you here methyltransferase 3 in green, CD3 in red, which is just a normal marker of T cells, and insulin in blue. And you can see that as the disease progresses, the methyltransferase 3 signaling intensity drastically decreases. And in the last stages of T1D, we virtually don't see any positivity, any nuclear positivity for methyltransferase 3. Well, one will say that we don't want to cure type 1 diabetes in NODs. We all agree with that. Even sometimes this is a challenge. So we had access to control and type 1 diabetic human islets. We culture them overnight. We pick up the islets that were really great with still some insulin granules, and we performed qPCR analysis. I'm showing here the T1D in red and the controls in gray, and you can appreciate how the methyltransferase 3 and then 14 levels are decreased in T1D compared to the controls. Well, this is a very dirty experiment because we know that we are comparing islets with many beta cells versus islets that virtually don't have any beta cells. So what we did after was to download again and reanalyze a single cell RNA-seq performed in islet beta cells from donors with type 1 diabetes, published by Professor David Arlen's group. I'm showing here the T1D beta cells in blue and the control beta cells in red, and the gene expression of methyltransferase 3, 14, and WTAP, another M6A writer. And you can see how all these different M6A writers show downregulation in T2D compared to the control beta cells. So what we are seeing is that in established diabetes, we have a clear downregulation of M6A writers methyltransferase 3 and methyltransferase 14. But in early stages of disease, we actually see an upregulation of the M6A writers and M6A levels. So the question for us was, how can we mimic this in vitro? Which tool can we use to study the role of M6A methylation in T1D onset and study what is this precipitating event which triggers autoimmunity? And we thought about pro-inflammatory cytokines. So what we did was to culture human islets in vitro, and then we challenged them with basal media or with different cytokines or cytokine combinations. And we used the best method to quantify the total levels of M6A. We performed mass spec. So I'm showing here the percentage of the basal condition of the M6A and adenosine ratio and the different conditions that we tested. And you can see that overall, every cytokine or cytokine combination increases M6A levels. But more specifically, the combination that further increases the M6A levels compared to the basal state is the combination with interleukin-1 beta and interferon alpha. And this is the combination we use to perform M6A sequencing. We further validated this, and so we culture human islets overnight, and then we challenge them with interleukin-1 beta and interferon alpha for 48 hours, and we compared these to a basal state. We embedded these islets in agar, and we did staining for methyltransferase 3. I'm showing here two different islet preparations on the top and on the bottom, and you can see the basal on the left and the cytokine treated islets on the right, and insulin iskostein in red, methyltransferase 3 in green, and DAPI, a nuclear marker, in blue. Well, the first observation is very clear. Looking even at the basal state, how the intensity of methyltransferase 3 is extremely heterogeneous between beta cells, even in a basal, normal, non-diabetic condition. The second observation that is also very striking and clear is that when you compare the basal state versus the cytokine-treated state, there is a clear upregulation of the nuclear intensity of methyltransferase 3 compared to the basal. So we were very confident that the cytokine combination was a very good tool for us to study the role of M6A in T1D onset. And so a question that we had before, particular from the most skeptical peers, is, well, you guys are floating human islets with cytokines. How does that mimic T1D onset? Well, I would just like you to show this very small piece of data. On the left, you can find a PCA plot showing the log fold changes versus the p-values of a microarray data from laser capture microdissected pancreatic islets of patients recently diagnosed with T1D. The first observation is there is a clear bias towards upregulation. And the second one is that virtually all of these genes belong to the type 1 interferon response, to the innate immune signaling. Now on the right, I'm showing you our own data, the input RNA-seq data that we use to normalize our M6A data on the human islets challenged with interleukin-1 beta and interferon alpha. You can see first the same bias towards upregulation in our treated islets. And you can see that we detect virtually every single of the upregulated genes in the T1D onset here on the left. So for us, this is very clear that this is a very good tool and that indeed pro-inflammatory cytokines, they do recapitulate the transcriptomic makeup of type 1 diabetes onset in human islets. We did M6A sequencing. I'm showing here a histogram where it shows the gene count, the number of genes versus the log fold changes. And consistent with the upregulation of M6A and M6A writers, you see an increased number of genes presented increased M6A peaks compared to genes showing decreased M6A peaks. And a very brief analysis that we did after was to intersect our M6A-seq data in human islets with the RNA-seq data of the same human islets plus the endo-CBH1 cells. So we are asking what are the pathways of the genes that are M6A altered in human islets and which gene expression changes both in human islets and in endo-CBH1 cells, a human beta cell line. And when we did pathway analysis, clearly the two main enriched pathways were related to defense response to virus and to the innate immune response. So we are actively working on these pathways, and we think that really healthy islets, healthy beta cells, when challenged an environmental challenge such as a virus, such as COVID, that has been now being shown that is increasing the incidence of T1D in some regions, are able to upregulate its M6A levels and adapt and survive. But a stressed beta cell by high levels of ROS or high levels of ER stress will present low M6A levels and will not be able to maintain this upregulation. And so this will lead to two possible outcomes. It will lead to an exacerbated innate immune response and cell death, or on the other hand, it might lead to an improper innate immune response and the self-recognition failure and autoimmunity. Well, going forward, we will have to face a couple of challenges. For example, we will have to really dissect, distinguish the primary versus the secondary roles, the secondary functions of M6A writers and erasers. Why this is important? Because there are now a couple of papers showing that some of these M6A writers and erasers are able to bind chromatin and modify chromatin accessibility. So one, we need to work out and really distinguish M6A specific and mediated effects versus chromatin mediated effects. Well, due to the limitations in our sequencing strategies, particularly the strategies in which we use antibodies to pull down M6A, we usually are testing and are analyzing the most abundant RNAs, like the messenger RNA. So as we improve our protocols, this will be extremely useful to test what is the role of M6A in small non-coding RNAs, in SNO RNAs, in long non-coding RNAs. And the last two challenges, and one leads to the other, is that clearly the M6A levels and the levels of the different M6A writers and erasers, they seem to be very heterogeneous across different cell types and across different tissues. So we will really we really need to work on really developing single cell strategies. And finally, when this sequencing era is over, we will have to focus on functional experiments. We have to be creative, and we really have to push our imagination. And I'm including here this painting because I think there is no age limit for imagination. This is a painting from Amadeu de Souza Cardoso. He belongs to a first generation of Portuguese modernist painters. And we learn about him a lot in middle school, in high school. But it took me more than 25 years to start seeing insulin granules in these wars. So we probably will have also to go back to our old data and revisit our old data on the basis of what we know today. So the last minute for the most important slide, the acknowledgement slide. So I would like to really thank Professor Kulkarni. He has been extremely supportive. I really remember the Tuesday morning in our weekly meeting that I knock on the door and I told Roy there is this thing about M6A. I really want to do M6A in type 2 diabetics and control islands. And he said almost immediately, oh, that's exciting, let's do it. And then I said, Roy, there's a problem. I think we need more than 20,000 islets per patient. And still, with such a financial effort, he said a strong and convincing, oh boy, okay, let's do it still. So I will also thank the help of our colleagues in the lab. We have really talented people in the lab, and I've been getting the help of them in many of the different techniques in which they are experts. A special thanks to our collaborators in Chicago, Professor E, Scott, and Jianbu. We have learned a lot with them regarding M6A sequencing and analyzing our data. Yeah, they also provide us plenty of knockout mouse models. Professor Décio has been fundamental in the cytokine treatments optimization. It's funny because every time I think I see a new gene in our own data sets or a new pathway, I Google it and I see Décio reporting this in the 90s. Another thank for our collaborators, Dr. Mark Atkinson and Clayton Matthews. They have helped us in sharing data, and through NPOD, they gave us some T1D, they provide us with some T1D sections. Dr. Al Powers, with the help of a Helmsley grant, provide us with the human type 1 diabetic islets and also age-matched control islets. Professor Wei Jun and Xiao Lu have been helping us with some experiments in proteomics. Audrey Parent has now been helping us in performance stem experiments in beta-like cells differentiated from stem cells. Thanking a last thank you for the different Jocelyn cores, and of course to NIH and EARN for their financial support in the form of a UC4 grant. ADA for sponsoring with ADA. And the Yacoca Family Foundation. Thank you very much. And I will be very happy to take some questions.
Excellent. Thank you so much, Dr. De Jesus and Dr. Kulkarni. Great lecture. Could we see if there's any questions now from the audience? Raise your hand if you would like to ask anything and go off mute. I see Dr. Ramiro Raghu. Would you like to ask a question?
Yeah. That was terrific, Dario Rohit. Really fascinating stuff. Can you guys comment on the specific number of M6A modifications per transcript? Does that matter? Or the location of those specific modifications? Does that alter the sort of the peaks that you see? And so could that matter?
Yeah, I think that's a good point, Raghu. I think the data are still emerging, but we can say that the predominant methylation pattern is in the 5' UTR and coding sequences, and approximately 3 to 5 sites per mRNA is the approximate sort of estimate of so far in the different genes. And as I mentioned, about 1/3 of the human genes, 7,000 genes, have been proposed to be methylated. And so there's a very large number which are important.
Great, thank you. Is there another question for our speakers? Yes, Dr. Khan?
Yeah, I just have a general question about when you discover the RNA modification, and what is the general tool you can study the function of the particular modification? Like, if you think like some of the modification is important.
Dario, you want to take that, or you want me to?
I'm very happy to take that.
Okay.
So, yeah, that's a very good question, Wei Jun. So these experiments are very tricky. So a way we try to study a specific modification in a specific transcript is by knocking down independently the different M6A writers. And so if knocking down methyltransferase 3 decreases the protein levels of the mRNA or the mRNA levels of a specific transcript, and knocking down independently all the other M6A writers, then for sure, at least this gives us a really high confidence that this effect is mediated by this specific modification and by M6A. So I think that's the way of addressing it. Another way of addressing it is, for example, by overexpressing catalytic mutant methyltransferase 3. And so then we will be able to really dissect the role of M6A in this target pathway or gene. So it's just a short answer is we have to really play around with a lot of knockdowns.
Okay, thanks.
Great, thank you. Dr. Blondell? Did you have a question?
Yeah, I was wondering. I'm sure it's not easy to measure, but I was wondering about the cell specificity of some of those modifications, and several of us have talked about the potential for detecting RNAs out of the cells, even possibly in the blood, to report on beta cell function. Do you see a time when a higher resolution besides just sequencing RNAs in the blood, but including M6A marks could refine this kind of analysis and possibly detect RNA that do come from, let's say, a stressed beta cell?
So maybe I could start and Dario could add. Olivia, we have been discussing this a little bit for the last 1 or 2 years, and the methodology is getting better. For now, I can say that if we have enough mRNA and enough dollars, we can sequence deep enough to begin to look at nucleotide resolution, which specific nucleotide is getting methylated. So it's called SAC-seq. That's the latest acronym for that. But it is possible. The challenge is to go a very high sequencing ability. That requires a lot of dollars, especially if you want to do, have replicates and technical replicates and so on. So it is getting there, and we hope that the next 1 or 2 years is going to get much better. But the second point you mentioned, whether we can look at these changes as biomarkers and circulating in the blood, that's a very exciting possibility. But the question for us is, can we begin to look at this in the context of extracellular vesicles, exosomes? Are those packages that are being carried by the exosomes, are they methylated, and can they be serving as biomarkers? And that is an open question that should be addressed.
Great. Dario, did you want to add to that?
No, I think Rohit was very clear. Yeah, the amount, the material needed for now is the greatest limitation. Yeah.
Thank you. Dr. Sayed, maybe one last question before we wrap up.
Hi, Dario.
I'm from Farooq from Indiana University. I have a question about, do you know like whether the methylation, N6 methylation affects the translation rate?
It does,
On
it
mRNA.
does, it does, it does. People have shown that by doing ribosome and polysome sequencing, and clearly M6A regulates translation efficiency.
Thank you.
And there was one question in the chat. Have you looked at methyl RNA patterns of exocrine pancreas from T1D?
Yeah, that is something that we are doing right now. As I'm sure Dr. Parekh will appreciate, getting high quality RNA from acinar cells is a huge challenge. So we're beginning to address this in different ways, you know. So it is in the works.
Excellent. Okay, thank you very much. We appreciate both of you and your wonderful talks today. And thank you to the audience for attending. Again, this is will be on video if anybody wants to look at it in the future. Thank you for joining. Bye-bye.
Thank you.